As the diversity of animal phyla rapidly increased during the Cambrian, the diversity of the Ediacaran life forms declined. So, what caused these trends? How is it possible that there was such a rapid proliferation of animal body plans in just a short 10 million years during the Cambrian explosion? There are three hypothetical drivers for the rapid evolution of animal body plans during the Cambrian explosion. The big one, remember, nature red in tooth and claw. The predator-prey co-evolutionary arms race. Just like between the Galapagos marine iguana and the racer snake. Fossil evidence indicates that during the early Cambrian period, predators acquired novel adaptations for catching prey, such as piercing mouth parts and claws. And then prey species acquired new defenses, such as protective shells on the trilobites, or you probably remember the spikes on hallucigenia. As new predator-prey relationships emerged, natural selection may have led to the decline of the soft-bodied Ediacaran species and the rise of the many bilateral phyla that we still see today. We still see predator-prey coevolutionary arms races unfolding today. So be sure and check out the video below on moths that have evolved the ability to jam bats' sonar. The second hypothesis focuses on an increase in atmospheric oxygen that preceded the Cambrian explosion. More plentiful oxygen would have enabled animals with higher metabolic rates and larger body sizes to thrive, while potentially selecting against other more sedentary species. And then the third hypothesis proposes that genetic changes affecting development, such as the origin of the famous Hox genes, incredibly important genes in coordinating animal development, facilitated the evolution of new body forms. More on evolutionary development, or EVO Divo, in a moment, but for now, it's important to point out that these various hypotheses are not necessarily mutually exclusive, meaning predator-prey dynamics, increased atmospheric oxygen, and changes in developmental genes, the origin of new Hox genes, each may have played a role in the explosion of animal body plans that we see during the Cambrian. Okay, what about these monsters? These surely must be Cambrian creatures from 525 million years ago. Nope. These are all creatures that we find among us today. The sand hopper is on the top left. This is triops here on the bottom left. Brine shrimp. They're sold as sea monkeys in the store. If you've ever cultured sea monkeys, they're triops. And then my favorite over here. This is a giant marine isopod. So that's a cousin to the roly-polies that you maybe caught as a kid. These giant isopods can grow up to 14 inches long and weigh, get this, 3.7 pounds. Could you imagine an almost 4-pound roly-poly? These animals have a lot to teach us about the underlying genetics that drove the evolution of animal body plans during the Cambrian explosion. Evolutionary developmental biology, nicknamed EVO Divo for short, is a relatively new biological discipline that compares the developmental processes of different organisms, looking closely at those Hox genes, that's H-O-X, Hox genes, to infer the ancestral relationships between them and how these developmental processes evolved. So I like to think about Hox genes as kind of like the conductor at the front of an orchestra. So the organism goes through a process of development. The Hox genes are very important and highly conserved genes, meaning they don't mutate often. They are going to coordinate the development of the organism segment by segment by switching on and switching off developmental genes. Similar to the way that the conductor at the front of the orchestra may start the cellos and then the violins and then pull up on the cellos, right? And so Hox genes are coordinating the development of each of the segments, whether we're talking about segments on an arthropod, a Drosophila fruit fly, or the segments on a vertebrate like a developing human. And many of them are shared between fruit flies and humans. That's how important these genes are. So, important point. Genetically modifying Hox genes can induce radical and rapid morphological change. For example, mutating a Drosophila fruit fly Hox gene can cause the organism to grow legs where antennas should be. So underlying mutations in these Hox genes are likely what drove the radiation of the astonishing diversity of animal body plans we see during the Cambrian, as certain species are going to develop antennas and claws and different types of legs, walking legs and swimming legs and anchoring legs, results in an incredible explosion of form.